People ask about the role of technology in life science. What really drives the latest discoveries in life sciences? Is it individual initiative? Is it a good idea? What causes the advancement? I think a lot of it is driven by technology. And sometime a technology comes along, and if you learn about the technology, you get ideas to use that technology to address questions that you maybe didn't even think about before because you just had this bias that, oh, that's not possible, so I won't even consider it. But the technology makes so many things possible that you can really be innovative and have no limits on the questions you're asking. So I think technology drives a lot of what happens in life science. The problem is technology is expensive. To innovate new technologies and to build new instruments and to validate with relevant biological materials is very complex and multidisciplinary and expensive. So not every lab, a lab might consist of 5 or 10, or a big lab, maybe 20 people, you don't have the budget to buy these high-end instruments that are required to do some of the experiments you want to do. So there's a tendency to have core facilities or shared facilities or national, regional scientific facilities. And I think SciLifeLab is a great example of that, where you have top-rate scientists running facilities that are revolving around specific technologies in the life sciences. In the case of our work with SciLifeLab, we of course were interested in this multiplexing proteomics technology, but also we're interested in mass spec technologies, mass spec imaging technologies, in structural studies. For example, if we get a complex of a receptor with a ramp, that's great. We can validate it using the Boolean. We can then take that complex and study it with mass spec imaging or mass spec. But ultimately, we'd also like to get high resolution structure of that complex. So all of these capabilities are in centers like SciLifeLab. Interestingly, I got a new graduate student who wanted to work not on one receptor, but on all of the receptors at the same time. And this was a sort of a visionary project. Her name was Emily Lorenzen. And I said, well, you know, normally we work on one thing at a time, we try to focus. But if you want to try to work on 10 receptors in a family or even hundreds of receptors, we have to rethink how we do this. We need to find some technology where we can multiplex our scientific work. Why don't you review the literature and see what kind of multiplexing technologies could maybe be applied to our interest, which is G-protein coupled receptors. So, and actually we both did this, and we compared and contrasted notes, we talked about it. And then Emily said, you know, I really think the best technology is this technology called suspension bead array. And we don't have one of those Luminex machines at Rockefeller that can do suspension bead array. And I said, well, maybe there's one at SciLifeLab, because I knew that SciLifeLab was gearing up with using the human protein atlas antibodies, gearing up to do multiplexing and detection of antibodies in patient samples and in serum and in different kinds of environments. Maybe they have something that can help us. And that research is what actually led us to SciLifeLab, because they had the machine that was ideal for what we wanted. We didn't have a relationship with them, but we reached out to them initially by email and then a visit. And we were connected to Jochen Schwenk there, who had worked with Matthias Uhlin on the Human Protein Atlas. And it was a great story because, of course, everyone wanted to work with the latest technologies, including us. And we ended up writing a proposal and present presented to them what we wanted to do. And initially they said, well, you know, we don't have a lot of experience working with membrane proteins. We're usually working with soluble proteins. And my response was, that's the reason you should do this project with us, because we have to figure out how to use these amazing antibodies that you have in the Human Protein Atlas for membrane proteins, not just soluble cytoplasmic proteins. So that was really the start of the collaboration.